This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 60, recorded on July 18th, 2013. Hi, everyone. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hi there. How you doing?
Welcome back.
Yeah, nice to be back.
You were celebrating your old department, right?
I was celebrating the 50th anniversary of the Department of Molecular Biology and Microbiology at Tufts Medical School, of which I was a founding father.
So you were the founding chair?
No.
Just a founding member.
I followed Ted Park of Park Nucleotide fame, who founded the department. He got me there. And then he decided he had enough after about 8 or 9 years, and I stepped in. And I chaired it for an obscenely long period of time, namely 23 years. Wow. But it was such a nice place, and my colleagues were so wonderful. And we ran it like a family. So my job was essentially being the foreign minister. I talked to the dean. I didn't make any decisions. I didn't. Anyhow, we had a great time, and we had marvelous people who are alumni, have gone on to great things. And when I got up and talked at the dinner, I said just one thing, that as I get older, it becomes more and more satisfying to see young people do well. It's always been that case, but as you get older, it really matters even more. And that's just a source of endless joy.
Great. Also joining us today from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent. Welcome back, Elio. We missed you on the last one.
Thank you.
But you're not in South Carolina today.
No, I'm up in Chicago. It's much warmer in Chicago than it would be in South Carolina. We have the ocean, even though that Chicago has the breeze, and it's quite toasty here.
Are you taking a vacation?
Yes.
A well-deserved vacation from copper.
Yes, a well-deserved vacation.
Listen, maybe before we get started, I would like to make a comment about the TWiM I missed. That was TWiM 59, right?
That's right.
A couple of weeks ago, you two discussed a paper from the Rohr lab at San Diego State, dealing with the possible role of phages as defense mechanisms in the body. And I must tell you that, let me tell you what happened. I started to listen with the intention of just getting into it, and then I would stop and do some of the work I had to do. As I was listening, I couldn't stop. I honestly think that you two did a job which is truly extraordinary. In fact, if any of the listeners want to hear, and haven't heard it yet, want to hear how a podcast in science should be done, that's it. If anybody is writing a PhD thesis on scientific podcasts, that should be the paradigm. You guys were clear, to the point. It was just, your interaction was beautiful. I was proud of you.
Thank you.
Both of you.
Well, thank you. Now we're blushing.
And now what can we do next?
I don't know.
It was a hard act to follow.
Oh, should we just stop? We'll just stop then.
It's after we finish podcast 60.
Hey, we're at number 60. How about that?
It's been a very interesting and enlightening road. We've read a lot of papers that normally aren't in my wheelhouse, and it's actually re-energized me and got me thinking about other things. The graduate students are terrified to see me come down the hall for fear what I'll ask them, which is good. I mean, it's how science should be. You read
you
papers.
Should like graduate students.
Well, it's not that. You really, when I was at Indiana, I was really lucky with the faculty faculty were always engaging the graduate students wherever they were. They were always asking them questions. In the olden days, it was called pimping them for information, but it was really to get them to be curious. And I think that's my goal in reviewing the papers, is not to, put out the facts, because anyone can go through the paper much more efficiently than I. But it's really to generate curiosity, to think about,
Good.
how we can really begin to move our science to the next level, and not just keep reducing and reducing to the minimal essential facts. It's really to expand on. Oftentimes today, papers are getting shorter and shorter because of the word limits. And I think podcasts can actually serve to expand the discussions and help drive an interest in the field. And the phage paper was especially good because it really begins to articulate how the microbial world is probably one of the oldest components of our innate immune system.
Yeah, that was very nice. Really
So
was.
you have friends at San Diego State, right?
I do. Yeah, well, I hang out there. I have an appointment there, and as well as at UC San Diego. So I'm all over the place.
So Stan Malloy is at San Diego State. Is that correct?
He is the Dean of Sciences, and he is a phenomenal guy.
And
Just
then
terrific guy.
Rower, one of the authors, is also there. Is that right?
Yeah, he's at San Diego State, and lots of other. I have friends in both places.
I also received a very nice email from Mary Ewell, one of the co-authors on that paper. She also liked how we did it. She said it was very engaging.
Let me tell you, that's high praise. I've worked with Mary quite a few years now on the blog, and she is a tough and determined cookie.
Well,
So,
I
she doesn't
think
give it away.
that I think that the good work makes for a good podcast. And we try and pick all good papers. I think they're all good, but some results are just amazing, and I think that's, if we can reflect our amazement, that makes for a good podcast. So,
Well, why don't we get into today's?
all right, today we have two papers, and the first one is a paper in PNAS. It's called Surface Display of a Massively Variable Lipoprotein by a Legionella Diversity-Generating Retroelement. And the authors are Arambula Wong, Medkar, Guo, Gingery, Shorney, Lou Day, Ghosh, and Jeff Miller.
I should tell you that I know the two senior authors. Parto Ghosh is at UC San Diego, and Jeff Miller is a graduate from our department at Tufts, and he showed up. By the way, Jeff is the current president of ASM, or was he?
He's
passed. He's passed.
He's past president of ASM and a graduate from my old department. So he graciously came, and that was just lovely to see him there.
Another funny story about Jeff Miller is he came to the medical university when he was an undergraduate to do summer undergraduate research.
You're kidding.
And I think he principally came to Charleston just to hang out at the beach, but at least that's what he told me. But he came and did summer undergraduate research at my institution. We had Jeff come and talk about this very topic of this paper a few years ago. We have what's called Student Research Day, in which we stop all classes for a day and students present posters or give talks. And there's generally a keynote speaker. And we had Jeff Miller as our keynote speaker one year. And when I invited him to come, he says, well, I did summer undergrad research there. And so he told a cute story about how he was, he must have been 19, 20 years old when he came to Charleston, before he met up with the great crew at Tufts.
Well, let me tell you why I picked this paper. It actually originated with the last paper we did on phages and mucus. There was a sentence in the paper which read, quote, "Phages in the human gut encode a population of hypervariable proteins. For 29 hypervariable regions, evidence indicated that hypervariability was conferred by targeted mutagenesis through a reverse transcription mechanism. And this caught my eye. I said, what is reverse transcription doing in bacteria? Because reverse transcriptase, of course, was first discovered in retroviruses, and it, of course, is an enzyme that copies RNA and makes a DNA copy. And apparently, it's doing something in bacteria. So I emailed Jeff. I looked up some of the papers, and I emailed Jeff Miller, and I wrote, I've been reading your PNAS paper on phage, the PNAS paper on phage adhering to mucus, and came across your 2002 paper on an reverse transcriptase activity in a Bordetella phage. What is RT doing in a phage? And he answered, they are called diversity-generating retroelements, and they function to introduce vast amounts of diversity into target proteins using a unique reverse transcriptase-dependent process. Repertoires of up to 10 to the 26th sequences. We originally found them in phage, where they diversify ligand binding sites of tail fibers, but they are ubiquitous in bacteria as well. And so he sent a few papers, one of which was today's paper. Surface display.
It's quite a remarkable story. I think you're going to say this sooner or later, so I'll sort of...
it's okay.
I'll say it now. This resembles and beats the diversity in the production of antibodies in mammals.
That's right.
Oh, yeah.
That's right.
Yeah.
So this is really an incredible story. It's a very unexpected and possibly very important finding. Is how I would describe it.
So it's not known yet if it's important? That's what you would say?
Well, yes, it's important, but even though you find these elements all over the place, they tend to be reasonably specific in their target proteins. And so it really depends to figure out whether these target proteins are involved. Here they are. I mean, having a different lipoprotein on the surface of a Legionella makes a huge difference, probably with regard to response to immunity and maybe white cells and who knows what.
Yeah.
So here it's very likely. In other cases, it's a case-by-case thing. I would put my money on it that this is important, but it has to be done. The work has to be done.
So as Jeff had alluded to in his paper, the previous diversity-generating retroelement, which we'll call DGR, was characterized in a bacteriophage of Bordetella. And now in this paper, one is characterized in the bacterium Legionella pneumophila. And these elements consist of a number of parts. All right, there is a reverse transcriptase, of course, which is an enzyme, as I said, copies RNA to DNA. And during the reverse transcription is when the diversity occurs. We'll talk about that in a minute. And then there are various structural determinants in this region that are important for the generation of diversity. There's a template for reverse transcription, which is copied. And then what happens during copying of this RNA template, every adenine residue in this template, it's called a template repeat RNA, every adenine can be copied into any of the 4 nucleotides, a DNA copy. So for every A, it can be A, G, C, or T. So you can see now where the diversity comes from.
The sort of massive mutagenesis, right?
Right.
It's extremely clever, especially if you're trying to avoid antibodies, because the diversity that you can generate is just profound.
Right.
It's greater than the one of antibodies.
It is incredibly greater. Now, most of
go
ahead.
Sorry. One point that I think you were doing that these elements were discovered in phages, and now this is the first report of a discovery in bacteria.
Correct.
So there is a point that's very important here, namely that they look exactly like what you expect from transposable elements, which are passed on by horizontal gene transfer. So these are not just elements that sit in one chromosome of a given bacterium and that's it. They're really quite mobile.
Right.
So they are, the evolution that can take place here is gigantic.
Yeah. It's interesting that the enzyme puts in any 4 nucleotides at every A. So there must be some structural feature of the enzyme that allows that, which, as far as I know, hasn't been figured out yet. So what it does, the reverse transcriptase copies this template repeat RNA and makes a highly mutated DNA version of it. And then this is inserted to the 3' end of the target gene, that is the gene that is going to be hypervaried, if you will, by specific signals, and that's called mutagenic homing. And this is basically a retrotransposition process. It's a one-directional, unidirectional retrotransposition that requires specific sequences. So now your target gene, has all this hypervariability at the 3' end, and of course many of the sequences are going to be non-functional or lethal in some way, but some of them will end up being functional and can then do whatever it is that they needed to do. In the phage, the Bordetella phage, where this has been studied, the target gene for this hypervariability is a C-type lectin, and it's in the tail fiber protein of the phage. So presumably this gives diversity to the phage in terms of attaching to different hosts. So those are the general features of this diversity-generating retroelement, and the key, of course, is this reverse transcriptase, which makes the diversity itself. So they found this sequence in a Legionella, and you can see it because it has a lot of the features of a diversity-generating retroelement. It has the reverse transcriptase. It has a target gene, which is called LDT in this case, which is Legionella. I'm searching for the name of it here. Legionella. I can't find it. You remember, Michael, what it's called? Legionella something.
Legionella determinant target.
Right.
So they don't really know the function of this gene, but it's clear that it has a sequence at the 3' end called VR, or variable repeat, which is known to be the target for the reverse transcriptase product. It has reverse transcriptase. It has stem loop structures that are known to be important for insertion. It has an accessory protein gene called AVD, and it has a template for the RNA, the tRNA template. And so this locus in Legionella has all the features of a DGR, if you will. So the idea would be that you could generate 10 to the 26 different DNA sequences of this protein LDTA, which would give you 10 to the 19th different proteins. I don't even know if you can imagine 10 to the 19th. It's almost Avogadro's number, right?
Right.
Yeah, it gives a whole new meaning to the word phage display.
Really? It's huge.
I mean, it's just absolutely mind-boggling. And, think about all the people doing drug discovery, and they have their arrays of chemicals that they're looking for new drugs. Here, the bacteria have invented it millennia ago. And, they're effectively getting around the best drug system out there, our immune system, by doing this. We're going to substitute any nucleic acid piece at this end that, is exported to the surface of the cell, that confers the phenotype to the organism that will then target it for destruction. And the interesting thing about Legionella is that, recall that it's a facultative intracellular parasite, and it likes to live inside our non-professional and professional monocytic type cells, as well as it lives out in nature in the natural copepods that are in water.
Amoebas. You mean amoebas?
Amoebas. So it's really optimally designed to be moved around. It's a really very clever bug.
Amoebas are extra body macrophages.
Right,
That's right.
extra body macrophage, or they were the original macrophage before we captured them.
Macrophages are intracellular amoebas.
Right. So by looking at the sequence of this LDTA protein, remember they don't really know what it does, they can make some predictions about it. They think that first they can tell that it's probably exported by a very specific secretion system called the TAT pathway, which enables proteins to go across membranes. It also has what's called a lipobox motif, and this is believed to mediate lipid modification and anchoring to inner or outer membranes. So they think that basically this protein is probably shipped to the surface of the bacterium. And so this is one of the things they look at later. So they do a number of experiments which are aimed at really comparing this DGR with the Bordetella DGR. All the elements required, are they similar in functions? The first thing they do is they ask, can this make mutations? So they use PCR assays to determine if, in fact, the RT makes mutagenic products from the RNA. And, in fact, they show that this RT can make adenine-specific mutagenesis, and the products home to the C-terminus of this LDTA protein, this so-called VR, Vincent Racaniello region, which is known in the Bordetella phage to mediate insertion, targeted homing or mutagenic homing. They look at some of these other structures in this region, these motifs that are important for the Bordetella activity, and they show that, again, they're similar. And their conclusion is that the Legionella and the phage DGRs operate in a similar manner using similar conserved sequences. Then they turn to this LDTA, and they ask, what is this? And they do a whole lot of very nice biochemistry, localization, and they basically show that this protein, LDTA, is an outer membrane protein, and the C-terminus is exposed on the surface. Now remember, the C-terminus is where the hypervariability is going to be introduced. So that's interesting that the variable protein is exposed on the surface of the bacterium. They also show that this TAT secretion system is in fact the one that is used to export LDTA to the surface. There's very specific genetic ways that they can do that. And they also show that the protein is modified on a cysteine by the addition of a lipid, and the modified protein is then placed on the surface of the bacteria and anchored to the outer membrane by this ASA-linked lipid modification. A whole lot of really nice biochemistry and cell biology here. So basically, this protein in this DGR, this LDTA protein, is an outer membrane protein of Legionella, and the C-terminus is exposed on the surface, and that is, of course, the hypervariable sequence. They do look at some other Legionella strains to see if they have this cassette as well. I guess there are over 50 species of Legionella, and they looked at 12 clinical isolates and found 3 that have DGR loci in them. So it's not in all of them, but it's in some, and they have similar conserved elements. And they think that these are moved around in modules from one species to another.
Right. So that this is they get amplified ecologically to many other organisms.
Right.
Yeah.
And they're also in dental bugs. If you look, they're in Treponema denticola, Bacteroides fragilis, which is the most common gram-negative below our diaphragm, Bacteroides thetaiodomicron, which is in our mouths, and an organism we're going to talk about, I guess, in the next paper, Schmidt.
Schmidt.
Yeah, Schmidt. And so, this whole business of the outer membrane of these gram-negatives being so important, and if you think about the oral flora, like treponemes, the treponema, of course, are one of the microbes associated with acute necrotizing ulcerative gingivitis, and you have this mixed infection of the oral treponemes, the oral streptococci, and then the stinkiest microbe out there, fusobacteria. all get together and they ulcerate your gums. They just wreak havoc in your mouth if you let them get out of control. And oftentimes we don't actually know what precipitates acute episode of Vincent's infection or ANUG. And it may be that these microbes just happen to be able to become locally invasive because of this RT effect, that they are just able to attach so much better to our gum line and wreak havoc, because they hit, if you will, the jackpot of the slot machine, shuffling this outer membrane protein.
Yeah. So that's really the issue here. We don't know what this LDTA protein is doing on the outer membrane, but it could help adhesion or invasion of some sort, and having variability.
Or protection from phages.
Protection.
Yeah. Can I ask you a different question? They did a fair amount of structural work on some of the proteins involved, especially this accessory protein. It's an accessory protein for the RT, for the reverse transcriptase. It's called AVD. It's nothing but beta sheets.
Beta sheets, yeah.
Vincent, you probably know something about reverse transcriptases in where they belong in retroviruses. Do they use an accessory protein similar to this?
No, not that I know of. No.
Just interesting.
So let's ask Vincent the next hardest question: is this the chicken, and the RTs found in viruses are the egg?
Yeah, well,
that's a good question.
Assuming a chicken becomes before an egg.
To us bacteria chauvinists who believe that everything was invented by bacteria.
I suspect there's a common ancestor of all of these, right? It's
Yeah.
probably a very old protein. Probably reverse transcriptase dates to the RNA world, I would guess.
Yeah.
So, as listeners may know, there's a theory that the earliest life on Earth was RNA-based, and eventually made a transition to a DNA world. And, of course, a reverse transcriptase would be the perfect enzyme to make that transition, right, to make RNA to DNA. So it could be that it's really old and was before, who knows how old. The RNA world is a very primitive cellular world. So it could be that, and then we don't know who evolved first, so...
Well, it also makes sense that to think that in early evolution, mutation rates must have been gigantic.
Yeah.
Biology was trying its hand.
That's right. That's right. Yes, so I like that.
Having a mechanism for creating so many different mutations may be a very handy thing.
Yeah, that's a good point. So maybe this is a descendant of the earliest RTs, which were very mutagenic, and then they evolved to be more sophisticated in viruses. Could be. Very interesting. The last thing I want to point out here is that this LDTA protein and similar proteins are apparently in these other bacteria, or at least some of them, is a C-type lectin. Lectin, of course, is a protein that binds carbohydrates, and C, I believe, means that calcium is required for the binding. And there are lots of different lectins that bind all kinds of sugars. So this gives us some suggestion about what this protein is doing. It may be interacting with a sugar or a glycoprotein of some sort. But the diversity, as Elio said earlier, they make a point of saying here, to our knowledge, the amount of diversity that can be generated by these DGRs is greater than for any characterized biological system, including the diversification of immunoglobulin scaffolds.
Well, can you make the case that this may have been the origin of the immunoglobulin system at a much, much, much later point?
Sure. The basic mutagenic function could be, yeah. Very interesting. So this is all new to me. I didn't know any of this. I think this is amazing.
Well, you've done pretty well considering your ignorance.
I'm very ignorant, but I try to fix it on a daily basis. Is there anything that I left out that we should mention?
Nope. I think we'll get a letter from Jeff if we did.
That's true.
If he listens, I don't know if he listens. All right. Our next paper is a Michael paper. which is...
I've been trying to get this one into the listeners, and this is sort of a tease before I'm going to bring one of my colleagues from the medical university who works in this field, who's done a fine piece of work in this. This is a review of what's going on in this emerging area of microbiology, and the title of the paper is The Conversion of Waste into Bioelectricity and Chemicals by Using Microbial Electrochemical Technologies. And this is by one of the leaders in the field, Bruce Logan and Cornell Rabney. If I butchered the last author's name, I apologize in advance. But this is a really neat paper because it's a primer for all of those of us who are interested in this new emerging field of how bacteria can do useful things for us, like make electricity. Most folks, when you ask them what microbes do, they just think they make yogurt and pickles, and they don't really...
Or make you sick.
Or make you sick. They don't really appreciate the fact that we can use them to generate many useful chemicals for the cosmetics industry. And in fact, a lot of the chemicals that are generated from this new and emerging field are actually taken directly by the cosmetics industry because they're so pure. They're much cleaner than the chemicals that can be generated from the petrochemical industry, where they have to scrub them to remove some of the impurities associated with them. And one of the first things that the authors tease us with is this fun fact. Treatment of organic risk wastewater currently consumes about 15 gigawatts, or 3% of all the electrical power produced in the United States.
Much of that is due to the need to aerate the sewage plant, the sewage, right?
Correct. And
that takes a lot of energy.
And considering what's going on the East Coast of the United States and the Midwest, and even the West Coast, we've been under tremendous heat. So we've been using a lot of electrons to keep ourselves cool. And these bacteria, as they begin to take us through the system, and they tell us that capturing part of the energy would provide a new source of electrical power, because, animal waste coming from us, whether it be domestic, industrial, or even the associated agricultural waste, if we could begin to harvest the energy associated with that waste material and convert it into useful things for us, whether it be something as simple as electrons that flow through copper wire into our homes to charge all our electronic devices, or whether it be useful chemicals such as what are used by the cosmetics industry or even the ethical pharmaceutical industry to make useful things, it really would be quite valuable.
Poop to scoop. I call it poop to scoop.
Yeah. And so the first thing they do is they ask the very simple question, and how do microbes generate electricity from organic material? Now, any student of microbial physiology knows the answer to this question, because the most pressing thing for the physiology of the microbe, no matter what it eats, is it has to regenerate oxidized NAD. Because it's the oxidized NAD, no matter how it generates its energy, is the chemical that takes that initial electrical hit. And when it takes that electrical hit or harvests that electron to do useful work by the bacteria, whether it be to make carbon, whether it be to make nucleic acids, proteins, whatever, it needs reduced purity nucleotides. And so it will take the oxidized form, and often it's NAD. And if you don't have oxidized NAD, you could have all the energy in the world, and the microbe wouldn't be able to do anything with it. So the first thing that the microbe has to do is to figure out how to regenerate that oxidized NAD. And, the rest of the most simplest system, of course, is the Embden-Meyerhof pathway, where we all understand from our early physiology is glucose will ultimately become lactic acid. And the reason we generate lactic acid, or the bacterium generates lactic acid, is so that it can reform that oxidized NAD to take the electrons. So now, if we think about why a microbe would want to generate electricity, it just wants to get rid of that waste electron. It just wants to put it someplace. And so that's what Logan and Rabi begin to take us through. It's talking about where to put the electrons and what to convert them into. And they begin to describe the world of a bioelectrical system. And so you have to go back to understand cathodes and anodes. So in the true sense, when a microbe puts its waste electrons into pyruvic acid to make lactic acid, the pyruvate is effectively serving as an anode. And the electron goes into pyruvate and it makes lactate. But wouldn't it be better if we could take that lactate and now convert it into something else where we split off even more electrons and move them in a circuit where they go from an anode ultimately to the cathode, and we effectively move the electrons. And so in this bioelectrical system world, you move the electrons literally in a wire. And so the electrons flow from the bacterium to an anode, and then they go from the anode through a wire, ultimately reaching the cathode, where they can be made into many different things, whether they be acetate, whether they be hydrogen gas, whether they be complex molecules like methane. Vincent lives in New Jersey, and the most famous methane-producing microbial system on the east coast of the United States is, of course, the Fishkill landfill. It's got lots of pipes shoved in it, and at night, when I used to drive across the Verrazano. That's it. That Italian-sounding bridge I could never pronounce. You could actually see the Fishkill landfill burning off the methane. And someone once told me it's one of the highest mountain peaks in all of New Jersey.
It is. It's actually Staten Island. So it's New York.
It's New York. It's on.
So the methane is being produced by microbial activity,
right?
And that's taking the electrons that have been generated elsewhere and converting them to methane, or using them to convert something to methane, right?
It's the ideal waste product because it goes off as a gas.
So in this anode cathode setup that you told us, if you don't take the electrons away from the cathode, they will just accumulate and you won't get any electron flow, right?
Correct. And then everything stops.
Okay.
It's all about electron flow.
Right.
And, this is one of the most basic systems. Recall back to when you took freshman chemistry and you learned how to do stoichiometry and you had to balance
God forbid.
You had to balance both sides of the equation. Well, that's effectively what's going on here. We have to do mass balance where the electrons have to go someplace, and if you add the electrons, and it's all these god-awful half-cell reactions that is sort of the last part of freshman chemistry that no one really quite understands because the freshman chem prof is rushing us through it. But that's effectively what Logan takes us through. He takes us through telling us how much energy that we can generate per meter square, and it's really pretty remarkable. They describe power densities, and power is, of course, nothing more than electrons because it does work. And, of course, you can energize the electron. That's what we do when we charge our cell phones, is we pump the electrons back into the battery to effectively energize it. And as we discharge our devices, any of you who have used a cell phone can appreciate, as the current flows, one of the byproducts of that electrical discharge is heat. You only have to talk on your cell phone for like 10 minutes or so, and you can feel it get warm. And that's because as the electrons are flowing from the anode to the cathode, you are effectively generating heat as it goes through the semiconductor processor to do all the wonderful things of a cell phone. So Logan describes power densities produced by Mfcs, which are microbial fuel cells, and he tells us they're generally lower than those using hydrogen fuel cells that we often find in today's hybrid vehicles, like, the Prius or the Ford Focus. All these hybrid cars that we see driving on our roads today use hydrogen fuel cells, which typically operate at temperatures greater than 200 degrees. And, short of spore formers, we know very few bacteria can live at temperatures in excess of about 42 degrees centigrade. So you, of course, can immediately appreciate how microbial fuel cells are going to be limited first by temperature. And then, of course, the hydrogen fuel cells make their living by flowing at very high ion densities. And, of course, in a microbial fuel cell, these ions can be both protons. And so as we would increase the proton concentration, we would lower the pH. And, we can only go down so low, or as pH goes down in number, of course, the concentration of hydrogen ion goes up and the cells effectively will pickle themselves. So you have this very fine line of what you can do.
Michael, you said 42 degrees, but aren't there ocean floor vent microbes that can live under higher temperatures?
Right. There are indeed. But, and in fact, when we bring my colleague, Hal May, he'll talk about how he's been using thermophiles to actually increase the power density. And so I didn't want to steal some of his thunder of talking about his thermophiles that he's isolated from, of all things, brewery waste. And where all good microbiologists go to find new bacteria, they go to their favorite brewery and ask them for some waste. And so back to the power density, water in most cases serves as the electrolyte for charge transfer in microbial fuel cells, as opposed to these high salt ion baths that are used in commercial fuel cells that are often found out there. And what I think the microbial community, the people working in this discipline, have done is they've been trying to figure out how to increase the power density while at the same time allowing fluxes in generating watts. And in fact, they described that microbial fuel cell power densities are much lower than hydrogen fuel cells, which produce the order of electricity as 1 watt per centimeter squared. And unfortunately, the bacteria don't seem to have the ability in order to get up that high.
So, Michael, in a microbial fuel cell, you have basically two chambers with an anode and a cathode, right? And
correct.
Then in the anode chamber, you have bacteria which are generating electrons by
feeding them to the anode,
which in that generation of electrodes is by breaking down sources, which would be
organic waste,
right? And the electrons flow into the anode; they go through wires, and you can tap that for making power, right?
Correct.
And then they go in the cathode, where they're then sucked off by other bacteria that are going to make things like methane to pull them away.
Correct. So the first simple fuel cells, the first simple microbial fuel cells actually just coupled the electron to oxygen that was around the cathode, and it just completed the half-cell reaction and made water.
So what kind of bacteria do we use in these fuel cells? All different kinds?
All different kinds. The most famous ones are Legionella and Geobacter. Geobacter is, of course, the favorite organism of another group, Derek Lovely, out of the University of Massachusetts at Amherst. And the Geobacter, you'll recall, we did that one paper on nanowires, where they have amino acids, typically charged amino acids, and the electrons move down these, if you will, pillin-based wires. And the electrons just bump along, very similar in concept to the way electrons move within copper wire. And, you know, it's really all about which microbes will function in order to give up their electrons and to these things. And once you can actually have a microbial community affiliated with the cathode, because remember, electrons are the currency of metabolism. And if you can capture the electron from that half-cell reaction, and in the early days, those electrons would be something as simple as adding an electron to a proton to make hydrogen. And of course, hydrogen is, of course, the bacterium's universal energy currency, because hydrogen, most microbes have the ability to convert hydrogen into useful electrical energy to effectively reduce NAD, to reduce purity nucleotides, which can then be used for biosynthetic reactions.
So are these all experimental, Michael, or are some somewhere being used to actually do this, to clean up waste and to generate electricity that can be used?
Lovely, of course, is working on microbial fuel cells that are actually powering buoys in the ocean to actually measure things like temperature and other things.
So these are low-power applications.
These are low-power applications, and it's now a blend of electrical engineers where you can take the electrons that are coming off of an anode and charge a capacitor and then discharge the capacitor down in series in order to effectively create the potential, because it's all about potential. Because remember, everything flows downhill. The waterfall metaphor truly works for power production because it has to perceive that gradient. And so you discharge the capacitor so it is at a lower potential so it can take more electrons, and then when it gets charged up, it discharges down. Because microbial fuel cells, unlike batteries, can't operate in series to generate more current. You know, just as you put two AA batteries on top of one another to generate 3 volts, you have your 1.5 volt battery plus your other 1.5 volt battery to generate 3 volts. Unfortunately, microbial fuel cells don't effectively work like that because the potential, the microbes are smarter than us. The potential isn't there. So the electrical engineers have played games with capacitors in order to lie to the microbe, in order to get him to do things. And so what Logan then goes on to describe is combining these microbial electrochemical technologies, which he abbreviates as METs, or MECs, which are microbial electrolysis cells. And he then really begins to talk about how to use bacteria in concert with reverse electrodialysis, which is a process used to directly create electrical power from a salinity gradient. So as you can well imagine, as the tide comes in New York Harbor, hitting the freshwater of the Hudson River, you could actually have pairs of ion exchange membranes in a stack to create a potential. And globally, you can generate they estimate up to 980 gigawatts of power harvested directly from a salinity gradient.
Which has nothing to do with microbes, right?
No, this is just, but he's talking about trying to understand how we can begin to have a favorable anode reaction provided by these exoelectrogenic bacteria, and that's the new term. We're looking for microbes that are extra electrogenic. They're throwing their electrons off of themselves and giving them up to an anode.
My take-home lesson, after trying to understand electricity, something I never understood, so I'm having a handicap here. But basically, it sounds like it'll be quite a while before one can think about large-scale applications of microbial generation of electricity, whereas on a small scale, there probably will be some applications. So it's a mixed bag. I think it's hopeful, but it's not something where you are going to be able to grow your culture of bacteria in the garage and generate electricity for the living room. Not for a while, anyhow.
No, but where I see it actually taking hold is in the developing world, where there's a pressing need for clean water, and here septic
which is low consumption, right?
Right. So here, the average septic tank, if you would imagine the waste from a community of, say, 20 apartments would come together, where you would get the scale and you would process the waste and you would convert it into something useful. Some of these valued-based chemicals that they're suggesting that can be produced, such as acetate, glutamate, propionate, or butane, and you may be able to even make fuel such as propane or butane that you could then use to cook, so you wouldn't have to use these charcoal fires. So you could really design a system. And I think what will happen is foundations like the Gates Foundations and these others that have been putting out calls to ask to operate toilets on a nickel a day, I think some clever engineering of communities where sewage and electricity to charge capacitors, to do simple things like charge cell phones, or light-emitting diodes to generate
run a small computer.
Yes.
Yeah, sure. I think you're right. I think that's likely to happen and probably happen soon. And so we're looking at sort of an incremental industry, really, I think. It's not like looking for cold fusion or something like that, which is probably not possible. This is most likely possible, but slower than we may like.
Yeah, and it's really going to create the marriage of the chemical engineer, the electrical engineer, and the microbial physiologist. Those three disciplines together will be able to optimize the system. And it's, the microbes at the cathode are really centrophs, where, the centroph breaks down the waste material, and then a synthetic microbe will build it up, and you can even generate methane off of this process in order to generate harvestable chemicals.
So do you always have to generate a gas, Michael? Because if you do, then I can't see this being in a battery-like container, right?
No, you can generate things like acetate. And that's what the cosmetic industry loves, is acetate and propionate and these.
So.
But don't you have to remove these from the cathode in order to allow new withdrawal of electrons? So you can't have a battery, in other words, like the batteries we're thinking of today. This could never look like that, right?
No, it could never look like that. You can cheat. Ethanol has a low vapor pressure, so if you're generating ethanol, you can just pass it through a heat exchanger and get the ethanol to vaporize off out of the system.
But then we'll all get drunk.
Well, ethanol is you can actually burn, and
So
ethanol,
but these, if you use this on a large scale, you can't. I mean, you're going to have gases and various things accumulating. You're going to have other issues, right?
yeah, you're going to have other issues, but that's where the chemical engineer comes in, because they can make these into useful products. And in fact, one of the best waste products to generate is, of course, hydrogen.
Yeah.
Because hydrogen is a motor fuel. You can actually compress hydrogen in the standard lawnmower engine without any substantial modification. You can just, feed it hydrogen gas and it'll blow up just fine. And the engines actually run cooler.
So basically, these require pretty industrial, large-scale applications where you can withdraw the products safely and use them elsewhere, right?
Correct.
It's not going to be in my laptop. A microbial fuel cell will never run my laptop.
Well,
I wouldn't bet on that.
No?
I wouldn't bet on that.
Your good friend from the New York Times actually wrote up one of Dr. Lovely's papers where he quoted Dr. Lovely, and he said that, you feed a little glucose into the beginning of your laptop and it'll run your laptop.
But the products, though, how do you get rid of the product? That's the, right?
It's the magic of chemical engineering. It goes back to the mass balance and the stoichiometry.
My laptop is going to be producing gas, is what you're saying, right?
Correct. Well, my laptop already, I went to work one morning and my laptop was sitting on my desk at home, and I came home and the laptop was at an unusual angle, and I looked at it. And what happened, it wasn't plugged in, is the lithium-ion battery burped hydrogen, and it generates gas all the time, I guess.
Wow, it's interesting. So do we need to find new microbes to do this? Is that what you were alluding to earlier?
Well, the microbes are there. It's these extra electrogenic microbes. They're out there in nature. And you can actually pull them off of the anodes, because they often attach to the anodes. They make great biofilms. And that's our friend Legionella. That from our last paper, Legionella is actually one of these exoelectrogenic microbes. And so how Legionella sticks to anodes, and the anodes are something simple, just like granulated activated carbon. Good old charcoal.
I was wondering if there was a possible connection between the two papers, and of course you found one, but there may be another one. The Miller paper might just be a way to do targeted mutagenesis on a different scale than we know now. And certainly if you have a good bug like Legionella, you can have a better bug.
Right.
Yeah.
By looking for it. And so I wouldn't be surprised that these two technologies will meld at some point.
Yeah.
Someday.
So this sounds like should be a really active area of research. Is that the case?
It absolutely is.
It is, and there are some companies that are making products based on this. So it's not all a pipe dream.
And of course, one of the best places to do this is at hog farms, because hog farms, the average human generates 250 grams of sewage a day, whereas the average hog generates 4 kilos a day. And so that's a lot of sewage treatment. And so consequently, in North Carolina and Iowa, there are more pigs in Iowa and North Carolina than there are people. And so consequently, these farms have their own sewage treatment plants, because they can't dispose of the waste just due to the biological oxygen demand, because it would overwhelm a sewage treatment plant or even overwhelm a septic field.
Yep.
This is great. This is a really interesting article. I encourage everyone to look at it. And I think this is great future for chemical engineers and microbiologists, right?
And that's, and there's some really neat basic physiology going on, and also the whole notion of how electrons move in such things as pili.
Right. And of course, we know that Michael's favorite things is electron movement.
It's the universal energy currency.
Right. All right. Thank you, Michael. I have a couple of emails I'd like to read before we wrap up here. Just 3. One of them is from Kelly, who writes, I'm reeling from this episode. That is the bacteria in the brain episode, I believe.
That was the good one.
The symbiosis, the web of life, the energies, chemicals, organisms of evolutionary progression shaping our world naturally and by human intervention with unknown consequences, then you all tie it to the brain microbiome alterations affecting behavior. Yes. A recent quote from a researcher says, soon we'll call our immune system the bacterial interaction system. Microbes rule our world and us. God is bug. Oh, the heresy. This is what we see with inflammation therapy. Bugs rule even if you can't culture them with Koch's postulates. I have a Talmudic question. If most alpha proteobacteria are phototropic and the brain doesn't get sunshine, does vitamin D endocrine system dysregulation have an effect on their population in the brain?
Holy moly.
Who knows?
You know, vitamin D is this emerging area of research all over academic health science centers because it's so important, and we know that humans make vitamin D from sunshine.
Right.
So, you know, he or she, if Kelly's a he or she, they may have something here. I'll have to ask my vitamin D expert at work.
All right. She sends a few links to papers on vitamin D, metabolism, and inflammatory and immune diseases, which we'll put in the show notes. The next email is from Joe, and this email was sent to one of our other podcasts, This Week in Parasitism, but I thought both of you would enjoy it immensely, as well as our listeners, so let me read it. He writes, here's a follow-up on your biofuels question. So in one episode of TWiP, we were talking about biofuels. To make fuels from crops succeed, we need a biotech breakthrough that I really thought someone would have done already. Basically, we need a bug that eats cellulose and converts it back to sugar so you can ferment it. Personally, I think there is a Nobel Prize in it for the group that creates an E. coli strain that converts cellulose to sugars. Once you have that, then you can feed the farm animals the corn and run your tractor on the corn stalks. Or you could use hay or grass clips or wood chips or waste paper, whatever is available cheaply. Until we get that bug, ethanol from corn will just be a niche technology. People will keep pushing to use corn crops or other high-sugar crops to make fuel, but the economics are not good and the lost opportunity costs are too high. Look how the modest current efforts in the U.S. have pushed up food prices and still required government subsidies to be competitive. No doubt there is a listener out there with lots of arguments for how great ethanol from corn is, but I don't see anything like the margin needed to make it a viable market-changing crude oil substitute. I will show my age and tell you that as a senior chemical engineering design project in 1980 at Purdue, we looked at how to convert crop waste materials like corn stalks into fuel, and it wasn't pretty. The only real way to break down the cellulose was to grind it up and treat it with hot fuming sulfuric acid in big reactors. Fuming sulfuric is 98% concentrated acid that is saturated with sulfur trioxide gas, brute force chemistry for sure. As I remember, you could get pretty good conversion of the cellulose, but the ugly part was separating the good stuff from all the waste acid and the non-reactive lignin. Once you got all the acid out of the good stuff, then you still had to ferment the sugars. It's not surprising that you don't see anybody running this process to make fuel. We need a biotech solution to break down the cellulose without all the mess. I think folks were looking at the microbes in termites' stomachs as a place to start, but I have not heard of any progress on this in several years. I will add that biofuels are not the only option for our fuel supply. For the past 100 years, we have had repeated dramatic reports that we are about to run out of oil, and yet it never seems to happen. I remember a particularly detailed one in Scientific American about 10 to 15 years ago with beautiful graphs and everything. Each time the trumpets of doom sound, some smart engineer or geologist comes up with a new way to extract more oil. I don't see any reason why this trend will suddenly stop this time. We still have lots of tar sands, deep oil, and shale oil that have not been touched. Please note that I am not expressing a political view on the social correctness of these options, just the technical aspects. Even more impressive are the reported quantities of frozen methane hydrate clathrates on the ocean floor that would likely be fairly easy to extract. Some estimates are that there are more than 10 times the amount of energy stored there than in all the oil we have ever used. Obviously, none of these fossil fuels address the CO2 generation concerns that many people have. Wind, solar, hydro, and even nuclear power all have their places, and I hope their niches keep growing as the technology improves, but nothing comes close to competing with chemical energy as a cheap, portable, high-density source of energy. One just needs to look at biology to see the truth of this. Plants fix the sun's energy into chemical forms that then cascade through the food chain, ever evolving into more complex forms. How cool is that? Thanks for helping me stretch my brain each week. Thus ends This Week in Chemical Engineering. So,
well, I think we need to send Joe a link to Logan's paper, because I think the paper we just did today fits his bill, because that's exactly what's going on. Is the microbes are taking waste and actually converting it into something useful. And if the motor fuel that it's able to generate, such as hydrogen or methane, can come out to the level and extent that Logan is actually talking about, though I'm in Elio's camp and thinking that we need to figure out how to scale it more efficiently, because I think it's going to be low scale and until we actually do some of the engineering. So Joe may be able to help Logan tool it up.
So cellulose to sugar, is that the key, Michael?
Yeah, cellulose to sugar. You take the cellulose, you make electrons, and then you make sugar at the other end of the cathode.
So we can't do that yet. Is that correct?
No, we can't. That acetate is a sugar after all.
Okay. All right, our last one is from Robin, who writes, how about phage fossils? Now that we know that phages have an apparently symbiotic relationship with mammals, is it too far-fetched to imagine that some phages were once incorporated into and expressed in metazoa?
Love that. Of course. Not so easy to prove, but,
Yeah, you could easily search the sequences in the database, right?
Well, what sequence you're going to look at? You want fossil sequences, and there are a few, but not many. And I don't know how easy that would be. But yeah, the point is well taken. I think she's right.
Best we have is to go, take a page from Michael Crichton and go into amber. And we already isolated the one microbe out of amber that was, 250 million years old. So the next question is, could we isolate phage out of amber? And with deep sequencing, we may get something 250 million years old. How old is the metazoa?
You remember not long ago we did this paper on how our gut microbiome acquires new genes to digest, right? Carbohydrates, right?
Like algal genes.
Algal genes. So you can search the human. Can you search the human genome and look for acquired phage sequences? That's the question. I'm sure this has been done.
Well, there's a big effort in trying to determine how can you tell viral sequences from non-viral sequences, and it's all over the place. I mean, there is a lot of work along those lines. I don't know how good the algorithms are right now, where you can simply go to the internet and figure out which genes are which, but sooner or later that will happen.
Right.
It's not impossible.
And then he finishes, the reference to salicylates in my last email was prompted by Dr. Schechter's comment that methyl salicylate reminded him of acetylsalicylic acid, and to show how the acetyl group was important in the clinical effects of the latter. Thanks for such thought-provoking and paradigm-shifting discussions.
I don't know about paradigm shifting. I once wrote a piece about paradigm shiftlets.
Yeah.
Not everything that's tremendous in our mind is necessarily. The definition of paradigm shift by, what's his name? Who was it that introduced that?
It was an ecologist.
It was, I'll think of it in a minute. Anyhow, it's a fairly rigid definition. It requires the total unpredictability of the new situation, of the new insight. And it shouldn't be it shouldn't be synonymous with great new experiments.
It's Thomas Kuhn, right? We both found the same post, right, Michael?
Yeah, probably.
I have to read that book. That sounds great.
Yeah, it's an important book.
Did you read it, Elio?
No.
The structure of scientific revolution.
I read excerpts of it. Let's put it this way. But it's just a very useful concept, and it's nice to keep it a little bit clean.
All right. That will do it for TWiM number 60. You'll be able to find this episode and all the others at iTunes and at microbeworld.org/TWiM. And we do love to get your questions and comments. Send them to twim@twiv.tv. Elio Schaechter can be found at the blog Small Things Considered. Thanks for joining us today, Elio.
Oh, such a pleasure.
Always good to have you. And Michael Schmidt, normally at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent.
Love the batteries. Just love it. Great stuff.
Batteries are one of the first things that probably every sixth grade science student has done, made the potato battery.
That's right. So how does that work, Michael?
It's all about electrons and flow.
Very good. Potato battery. I am Vincent Racaniello. You can find me at virology.ws. Many thanks to the American Society for Microbiology for supporting TWiM, and to Chris Condian and Ray Ortega for their behind-the-scenes help. The music that you hear at the beginning and end of TWiM is composed and performed by Ronald Jenkins. We use it with his permission. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.